Good morning, everyone. Myself is Dr. Aishwarya Kopulwar from the Department of Biotechnology, Priyadashini College of Engineering, Nagpur. Today, we are going to discuss about the polymerase chain reaction, commonly known as PCR. PCR was invented by Mullis and his colleague in 1983. He acquired the Nobel Prize in 1993 for this discovery. It has become the most widely used nucleic acid amplification technology and gold standard for amplification process in diagnosis and research. Why this is called as the polymerase chain reaction? The first letter polymerase is because only enzyme used in this reaction is the DNA polymerase. Chain because the product of the first reaction becomes the substrate for the next one and so on. The reaction components becomes in a number of quantities, so they are the target DNA which contain the sequence to be amplified, pair of primers, oligonucleotide that define the sequence to be amplified, DNTPs containing DATP, DGTP, DCTP and DDTP, which are the building block for the DNA synthesis. The thermostable DNA polymerase, which the enzyme which act as a catalysis for this reaction. Mg positive ions act as a cofactor for the enzyme DNA polymerase. Buffer solution maintain the pH and ionic strength of the reaction which is suitable for the activity of the enzyme. Let us discuss more about the primers. The primers are the short single-stranded DNA molecule. They are manufactured commercially and ordered to match the DNA sequence. Primers are the sequence specific. They will bind to a particular sequence in the genome. You are designing the primer if of a longer length, they becomes more selective. The DNA polymerase requires primer to initiate the replication process or also known as the extension of the primer. The tag DNA polymerase is a gram-negative rod-shaped bacteria and obtained from the hot spring. The template used for the PCR is generally the DNA, and DNA can be obtained from the dried blood, semen stain, vaginal swabs, single hair, fingernail scraping, insects in amber, Egyptian mummies, buccal swab and toothbrushes. The major contamination while isolating the DNA is of RNA, and that can be removed by using the RNase as an enzyme. The instrument which is used for the PCR is PCR tube having the volume 50 to 200 microliter. Thermal cyclers you can be used of different manufacturing companies, and the agar gel electrophoresis which is used for the visualization of the PCR product. What happens in this PCR reaction? The sample DNA, nucleotide, DNA primers and the thermostable DNA polymerase placed in the PCR machine. The double-stranded DNA are get separated by heating by 95 degrees centigrade. The mixture is cooled down to the annealing temperature which is different for the different primers. And the mixture is heated to 72 degrees centigrade for the replication, or it is also known as the primer extension. And the cycle is repeat many more times. The cycle may be repeat up to 20 to 50 cycles. So let us discuss what is exactly happened with this PCR technology. Double-stranded DNA is get denatured at 95 degrees centigrade into a single-stranded DNA. The temperature is lower down to the annealing temperature according to the primers. The primers are attached and the temperature is raised at 72 degrees centigrade, and it containing the DNA polymerase, DNTP, Mg positive and the buffer. And this will be the end product of the PCR reaction. The PCR product are the amplification of the DNA in an exponential way. A one copy of a DNA after 20th cycle, you will get a 20 lakhs copies of that particular fragment. In this picture, the first gel, you will be observing about the DNA visualization with the help of ethidium bromide, and this picture is documented on the gel doc system. Whereas in the next gel, you will be observing only 500 base pair amplification, which is specific to the gene, is obtained after the PCR reaction. Now coming to the applications of PCR, molecular identification, it can be used for the archaeological studies, epidemiological studies, then the molecular ecology, DNA fingerprinting, classification of organisms, genotyping, prenatal diagnosis, mutational screening, drug discovery, genetic matching, and the detections of various pathogens. It can also be used for the sequencing process, which is useful for the bioinformatics studies, genomic cloning, and human genome project. Lastly, but not least, it has been used for the genetic engineering in the site-directed mutagenesis, which is helpful for the proteomic studies and in gene expression studies. Thank you.